Welcome everyone to the IIVS-sponsored webinar on current perspectives and new directions for the bovine corneal opacity and permeability assay, concentrating on applications in the regulatory arena. I'm Amanda Ulrey, your moderator and webinar organizer. We will begin the webinar with a brief history and introduction of the assay, followed by a discussion on histology by Hans Rabi of IIVS. This will be followed by an introduction to the exam background review document, assay instrumentation, and results for specific classes of chemicals by Dr. Susanna Culla of BASF. Hans Rabi will end the presentation with information on the use of the BCOP assay for antimicrobial cleaning products. We'll close the webinar with approximately 15 minutes of question and answer with the two presenters. Before we start, let's take a moment to ensure that everyone is ready and familiar with the webinar control panel. Due to the large number of attendees, all audio other than the panelists has been muted and will remain so for the duration of the webinar. You may ask questions by using the question feature at the bottom of the control panel to the right. Click on the arrows to maximize the control panel and view the questions window. Technical questions can be submitted anytime, but will be held until the end of the presentation when they will be posed to our panelists during the 15 minutes allotted for the question and answer period. Any questions that are not able to be answered within the scheduled webinar time will receive responses from our panelists and will be distributed to all webinar participants along with a PDF of the presented slides. Lastly, we are recording this webinar and the question and answer session at the end. Let's get started. Today's first presenter is Hans Rabi, Vice President and Director of Laboratory Services at IIVS. Hans has been Study Director for BCOP studies since IIVS's inception in 1997, has served as an expert on several OECD test guideline panels, including the panel charged with drafting the BCOP test guidelines, and has been an invited presenter of the BCOP method at exam workshops and numerous industry-supported venues. Hans?
Thank you, Mandy, for the introduction. Okay, so as Mandy had mentioned, I'd like to go through a brief historical on how the assay came about, and then go through a fairly stepwise approach to how to conduct the assay. And our focus will be to identify the various steps that are identified in the OECD guidelines, but there are a number of cases where modifications and customizations to the assay can be conducted, and so we'll touch on those topics as well as we go through the presentation. So briefly, the assay is based on the use of bovine corneas for studying corneal damage. Some of the work has historically been done for looking at mechanical wounds and also chemical wounds. At Merck Corporation, Pierre Gautron and Josina had begun to utilize excised bovine corneas primarily using an opacity endpoint, much as it is conducted today, but have found that for some chemicals that the inclusion of the permeability endpoint was quite useful also. So the current methods have actually been used now for nearly 20 years. And their goals were essentially to develop the assay for personal protective equipment definitions for worker safety purposes. And their goals were to have this assay useful for a wide range of chemical and physical properties of chemicals. They were certainly interested in a very conservative assessment. And of course, they want to conduct this work by eliminating the use of animals for the evaluation of eye irritation. So there are several endpoints to the assay. The two key assay endpoints are both quantitative. The first is the measurement of changes in light through the cornea, and this is termed opacity or changes in opacity. And this is quite important, as changes in opacity are probably the key, most important event that may occur in eye patient because of its impact on visual acuity. Opacity changes can come from denaturation of proteins in the cornea, disarray of the stromal elements or other components that affect light passage through the cornea. The second endpoint is a measure, again quantitative, of the penetration of fluorescein through the cornea. And this measures changes in the barrier properties of the corneal epithelium, so that as chemicals break down this corneal epithelial barrier, then we would likely measure increases of fluorescein penetration through the cornea. And thirdly, we've worked quite a bit with utilizing histology on the treated corneas to assess the degree and the depth of the injury, and also to look for other potential cellular or architectural changes that may be indicative of eye irritation that perhaps the opacity and permeability endpoints alone did not pick up on. So now we'll go through a slide-by-slide of the various steps in the assay, starting with the receipt of corneas. And this is very well detailed in the OECD guidelines on the receipt of corneas. The goals really are to maintain the corneas fresh after collection from the abattoir, and the methods that we utilize are to place the eyes into a Hank's basic salt solution, typically done with antibiotics, and kept cool on wet ice until it's transported to the testing facility. Now, the individual eyes are inspected for corneal opacities and scratches, and this is something that each laboratory must do carefully because this is the first step for a true quality control to occur, and we recognize that at least 30 to 40% of the eyes that we receive are just simply not qualified for use in an assay due to these corneal scratches and opacities. Excising the corneas is done typically in a teamwork fashion. We have a team of biologists that will cut the cornea through the outer scleral edge with a scalpel and then remove the eye, excuse me, the cornea, by cutting around the outer edge, leaving roughly a 2 to 3 millimeter ring of scleral tissue on the outside. And we'll explain in a second why that's important to maintain that outer edge. So handling these corneas is quite important to handle them delicately and gently. Now the scleral rim, as you can see in this upper picture, allows one to hold the cornea with the forceps without actually damaging the clear portion of the cornea. The cornea is removed from the eye. The iridial tissue is carefully removed from the cornea without stretching the corneal tissue. And these corneas are then placed into a HBSS just prior to mounting onto the corneal chambers. This is a picture of mounting the corneas onto the chamber with the endothelial side down onto the rubber O-ring. It's very important that one places the corneas gently onto this corneal chamber and not drag the cornea across the chamber to avoid damaging the very sensitive endothelium. So, quick brief on the corneal holders. As you can see here, the chambers are essentially composed of two chamber halves, front chamber and the posterior chamber. They have glass windows which allow one to pass light through the corneas and measure the transmission of light through the chamber holder to the back side of the chamber. These chamber holders also allow one to fill media into the chamber holders to bathe the corneas in media during the treatment period. Now, one has to be quite careful in assembling the corneal chambers. Once the cornea has been placed onto the posterior chamber, then one would carefully place the anterior chamber holder onto the cornea and carefully hold it in place and screw the two components together carefully to avoid dragging the cornea or misshaping the cornea during the assembly step. Here's a picture of our colleague backfilling the chamber with phenol red-free medium. And the goal here is to fill the back chamber first to provide a similar type of hydrostatic pressure that maintains the cornea in its normal convex architecture. After the posterior chambers are filled, then the anterior chambers are filled with media as well. And it's very important to avoid generating any air bubbles or foam, as they may have deleterious effects on the cornea, as well as affect the opacity and permeability endpoints. So we place the corneas in the incubators for at least an hour, and this allows them to come up to normal temperatures. These cells are quite viable, and so the endothelial cell pumps, for example, are quite effective at maintaining hydrostatic control of the stroma. So we place them in the incubator for 1 hour at 32 degrees. It's important to maintain all of your equipment throughout each of these assays to precise specifications as outlined in the OECD guideline. The opacity is the first endpoint to measure, and we take an initial opacity measurement using an opacitometer, which is very simply a meter system which allows one to place a cornea into the light path. The light source passes light through the cornea. A light detector measures that light transmission and gives a very numeric opacity value on the opacitometer. This is a unit prepared by Spectro Designs. This is the standard by which the assay has originally been conducted. And with the opacitometer, we have 3 calibrators, essentially 1, 2, and 3, which give us opacity units in a linear range from 75 to 225 opacity units. Now, in the development of the OECD guideline, it was also very important to be able to have at least another alternative opacitometer which could be readily available. And so BASF, their engineering group, had built a opacitometer to provide precisely the same type of endpoint measurement as the existing standard, the Spectro design. They've built this to be readily available to the marketplace. They have also built a series of corneal holders to, again, meet the OECD guidelines specifically. And Susanna will give a bit more detail on this equipment in her presentation a little bit later. So the initial opacity measurements are taken on the corneas. After calibrating the opacitometer, we place the cornea with the cornea holder with the corneas into the opacitometer and take the initial readings. And the OECD guidelines specify that we should not utilize any corneas with opacity values, initial opacity values greater than 7. In effect, in screening the eyes ahead of time and looking for corneal scratches, we can readily identify corneas that would not pass that specification. So our typical initial opacity values range from, say, 1 to 4 opacity units. Now here are the parameters for the assay itself. And the OECD guidelines specifically identify that liquids are generally tested neat. So the idea here is that this is a test system that allows you to apply liquids as if they apply the liquids to the corneal test system in the same fashion that could potentially be exposed in vivo. Now, there are some materials that we may want to dilute ahead of time, and typically surfactants may fall into this category where there are a number of protocols that utilize 10% dilutions of surfactant with water. There are reasons for doing that to compare against existing in vivo data, or their goals may be to obtain greater resolution among test materials to compare various toxicity endpoints. For solid materials, these are generally tested as 20% suspensions, as the suspensions are typically prepared in water or saline. Now, for aqueous soluble materials, one can imagine that these typically form true solutions, whereas for hydrophobic or aqueous insoluble materials, a 20% suspension can actually be quite viscous. Now, for the exposure times for the test system, liquids are treated for a classic 10-minute exposure time. There are times when longer exposures might be useful, for example, for very dilute surfactants, or in cases where in testing very mild materials, one may want to gain increased sensitivity to compare among very mild formulations. One can extend that exposure time. We have a number of protocols allow for 30 to 60-minute exposures. Again, these are outside of the definition of the OECD guideline, but are more typical for a product development application. There are also cases where multiple exposure time protocols may be useful, particularly for looking at a time course of events. And there are several cases where we've utilized 3-minute exposures, the standard 10-minute exposures, or perhaps longer exposures to look at the kinetics of the eye irritation potential. In particular, we found that the 3-minute exposures are particularly useful for organic solvents such as small-chain alcohols and ketones, which have been identified as frequently being overpredicted using the standard 10-minute protocol. And the solids, which are treated as a 20% suspension, are classically tested at a standard 4-hour protocol. So here are some steps on dosing. The 750 microliters of a non-viscous liquid can readily be added to the corneal holders using a standard micropipette. And so what we do in our laboratories classically place the dosing solution into the anterior chamber and rotate the chambers forward, so that upon dosing and adding dosing solution to each of the chambers, we can then rotate all the chambers together as a unit to begin and initiate the exposure period. And rinsing after the prescribed exposure time is done by carefully aspirating the dosing solution up the anterior chamber of the cornea, adding media to begin the rinsing process, as you see in the top left picture here, swirling the chambers to help effect a bit more of the rinsing, and aspirating that rinsate. And we go through the same cycle, at least 3 cycles, and monitor the removal of the test material to assure we get a good removal of test material. Phenol red is used in the medium to monitor for changes in pH. It helps us observe how well and effectively we're removing test material. And after 1 rinse with the phenol red free media, then we'll refill the chamber with phenol red free media, aspirate, and then refill the chambers fully with phenol red free media. And as usual, we want to assure that we have not included air bubbles in the media. The holders are then placed back into the incubator for a post-exposure incubation time. In this period of time when the changes that are not immediate in the corneas may be allowed time to express. One can take an opacity reading at this point, but this opacity value is used diagnostically to look for the time course for opacity changes, but it's not used in the calculation of the in vitro score. Now, the second component of the assay is this post-exposure or expression incubation time frame. And for the liquid protocol, we typically have a 2-hour post-treatment incubation time. And this allows for expression of corneal changes to occur. For example, degradation of corneal architecture, loss of barrier function, epithelial damage, perhaps activation of proteases which allow for the cornea to become further degraded. We also can measure stromal edema as a result of the epithelial barrier loss. And this is frequently a time-sensitive event that takes up to 2 hours to occur. We've also found that for some chemicals which have a delayed effect in vivo, it's quite useful to have markedly longer post-exposure expression periods. This, again, is outside of the guideline specification in the OECD guideline, but is very useful for looking at changes in the corneas due to reactive chemistries, peroxides, bleaches, and other chemicals, where it's recognized that the corneal changes may take several hours to occur. The post-expression incubation time that we've typically used are up to approximately 20 to 24 hours, which is essentially the limit of the time frame that we can maintain viable corneas in these isolated corneal holders. One of the endpoints that's particularly useful here is to utilize the histological evaluation to look for cellular changes that might be indicative of downstream corneal degradation. Now, the final opacity measurement is taken after the post-treatment incubation period, and this is the value that we utilize for measuring the net change or increase in opacity due to the chemicals in the calculation of the in vitro score. Again, it's very critical to avoid having bubbles in the chamber as they can affect the opacity measurement. So the second endpoint after the opacity measurements are conducted is to measure the change in the barrier function of the cornea using the fluorescein penetration endpoint. And we typically apply 1 ml of fluorescein solution to the anterior chamber. There are 2 concentrations that have been specified, one for solids, one for liquids, fairly similar formulations. And in unison, each treatment group then is initiated by rotating the chambers into a horizontal position. The corneas are placed in the incubator for 90 minutes. And at the end of the 90-minute period, we can sample the posterior chamber for the transmission or penetration of fluorescein. In these pictures, you can see that the sample is taken from the posterior chamber is quite fluorescent green colored, clearly indicative of a corneal, a degraded cornea. Now, we want to collect the full volume from the posterior chamber because it's very likely that any fluorescein that's passed through the cornea has not diffused homogeneously into the medium in the back chamber, in the posterior chamber. So we collect the samples, transfer them into labeled tubes. We take the whole 5 mils from the posterior chamber. And again, we must mix that sample media very well so that we have a good homogeneous mix. In our hands, we typically use a 96-well plate reader to measure the fluorescence or the optical density of the fluorescein samples. The assay was originally established using a spectrophotometer with 1 centimeter path length cuvettes. As I mentioned, we use a 96-well plate reader, and we found that the specific type of plates we use at a 360 microliter volume is the appropriate volume in the 96-well plate to replicate the 1 centimeter path length. I think each laboratory that's going to conduct this needs to verify this using their specific 96-well plates, because each plate has slightly different volumetric configurations, and so one needs to verify that the path length through the plate is equivalent to a 1 centimeter path length. And we then measure the optical density of these samples, the fluorescent samples or fluorescein samples, at a visible wavelength of 490 nanometers. Depending on the equipment that you utilize, the linearity of the plate reader may be limited, and we have found that with our equipment, we have a linearity up to 1.5 optical density units, critical that each laboratory that conducts this work verify using a fluorescent solution series, verify what their linear range is for their equipment. Any samples that we have that are greater than 1.5 optical density values need to be diluted and reread, and that dilution factor then applied to the final calculations. Okay, now for the third endpoint, we'd like to discuss briefly collecting the corneas and fixing them for histological evaluation. Histological assessment is done to look for tissue lesions, and it's clearly become a very important part of our program with a number of clients. What you see here are a series of cartridges designed to hold the relatively large bovine corneas. We place a very soft sponge on the endothelial side of the cartridge chamber, so that when we place the corneas into the cartridge, we can protect the endothelium from further mechanical damage. It's important that we do this work quickly after the end point of the assay, but also extremely carefully. In this top left picture, the operator here is carefully removing the cornea from the corneal holder and avoiding stresses that may damage or further damage the compromised cornea. These corneas are placed into the chamber, into the cartridge, endothelial side down, and it's very important that this is done correctly the first time. One doesn't want to drag that cornea across the chamber to fit it in properly. It's quite important to minimize any handling artifacts on the corneas. Corneas are then placed into a fixative. 10% neutral buffered formalin is typically used. The classic histopathology then is performed after sectioning and staining using hematoxylin and eosin, so H&E staining, and our histopathology uses a top-down approach to look at the corneas from the epithelial side all the way through to the endothelium, and we evaluate for damage that may occur upon the application site and any damage that may be indicative of penetration of the cornea. So we look for both cellular and architectural changes in each of these cell layers, and also look for the degree of these changes to help us understand the nature of the injury and whether there may be a notable event here for irritation. So the next few slides are very simple chemicals used to look at changes in the epithelium in this case. SLS was used, with 1.5% SLS being somewhat typical of a number of personal care cleaning products. You can see that there's a degeneration of the upper epithelium, and so the two measures of opacity and permeability here show us some evidence of the event here. What's notable is that we have a beginning signal here of fluorescein leakage, which shows the degradation of the epithelium. In the next panel, we have further degraded epithelium due to a higher concentration of SLS, and so the fluorescein leakage is quite notable in this sample. We also begin to see evidence of stromal swelling as the opacity values have increased. In this case, in this next picture, we have an acid-containing formulation exposed for a relatively short time, but this cornea has been coagulated by protein precipitation, and so we see a quite high opacity induction here with a notable increase in vitro score. Here's evidence of collagen matrix swelling, which typically results in moderate levels of opacity. Case of a quite strong response from a peroxide-containing cleaner, where the peroxide had produced presumably outgassing of oxygen. And so we see extremely high opacity values in these corneas that have been quite swollen, and the normal architecture has really been devastated by the outgassing. And we also look for endothelial lesions, and we look for changes in the endothelium as a measure of a full thickness event where the chemicals clearly cause damage throughout the cornea, the epithelium, the stroma, as well as the endothelium. And those type of events would likely be quite severe and unlikely to be recoverable in vivo. I'd like to walk through the calculations for the opacity and permeability scores. And so the opacity is measured by taking the difference between the initial opacity value for your corneas, for your test chemical, for example, and measuring your final opacity value and calculating a net change in the opacity values. One does the same thing for the negative control, and one has an average negative control value. And this average negative control value then is used to correct the test material and positive control samples to get a final corrected opacity value or final net change. And then we take the average for each of the treatment groups. The permeability assay endpoint is handled very similarly, in that we take the fluorescein leakage values from the plate, we subtract the plate blank. If there are any dilutions that were made because the signal was above 1.5, then we would make a 1:5 dilution and reread. And as you look on this graphic of data, the first cornea, had an initial reading of greater than 1.6, so we took a 1:5 dilution of that sample to maintain the signal within the linear range of our plate reader to get a final corrected value. So in each of these cases, the negative control data are corrected or used to correct the test material and positive control data to obtain the final positive control and test material results. And one utilizes this then in a prediction model to calculate an in vitro score. And the standard in vitro score developed by Merck using a large set of their pharmaceutical intermediates was to take the opacity value plus 15 times the fluorescein leakage value to come up with an in vitro score. Now, we recognize that some surfactant classes, particularly anionic surfactants, tend to have relatively low opacity values, and so we focus primarily on the permeability scores alone to evaluate the potential irritation of those type of formulations. Those samples typically are not evaluated using this predefined prediction model. And here's the prediction model to categorize the irritation potential based on the in vitro score. And Susanna Kalla will introduce a bit more on the OECD guidelines, but the OECD guidelines typically utilize the 55 and greater severe prediction for the prediction of severe materials. The lesser categories of moderate and mild have at this point not been adopted in the guidelines. So I want to make some last comments about inclusion, requirements of inclusion in the protocols. We typically always include negative controls and positive controls. And to do so, we obviously have to have acceptance criteria with the goals of defining a valid assay and defining the criteria for acceptance. The goal here, of course, is to have highly reproducible test results and very useful and relevant end results for the test materials. What's very helpful is to include some sort of benchmark or reference material to help interpret the test chemical itself. And those materials should certainly be taken from a product class that is very similar or the same as the test materials. And it's very helpful for those materials to have good quality in vivo data, particularly if it's some sort of human event type data to help interpreting the test material data. For any regulatory submission, full good laboratory practices compliance studies is a requirement. Now, the negative controls are used to account for nonspecific changes in the test system as well as in the assay execution of the contract laboratory. Examples of the negative control are typically deionized water or saline or medium, preferably using water or saline if that is the diluent used for a test material, if a diluent is required. Now, the negative control, as we mentioned in the calculations, is used to correct for the opacity and permeability values. So it provides somewhat of a baseline against which the test material and positive controls are compared. The positive control has an additional function that it ensures the response of the test system and ensures that this test system response is reproducible from trial to trial. And so it's extremely important to utilize a positive control, a standard positive control that is well characterized within that testing facility. We've classically used ethanol as the positive control for liquid test articles. Imidazole is used for solid test articles, or excuse me, 20% suspension of imidazole is used for solid test articles. The positive control certainly must be used with every assay to assure that the assay performance is acceptable on the day of the conduct. And an example of the issues that come about, and the only way to evaluate for changes in the test system is to include a positive control. So we have a defined acceptance criteria that the positive control must fall within a 2 standard deviation range of normal results. And this graphic here shows the responses, the historical range of responses for ethanol over a 5 or 6 year period. And it shows that the 2 standard deviation range has an upper limit and lower limit of roughly 39 and 66 here. There are a number of samples here that you can see have fallen outside that range. We recognize that there are some seasonal changes, so in the summer we tend to get slightly higher results, in the winter time a little bit lower. And clearly, had we not used a acceptance criteria for the positive control, or even conducted positive control in this case, we could very readily have had results for test materials that were likely to be over-predicted in this case, or, excuse me, under-predicted in this case. All right, so I'm going to pass this back over to Amanda Ulrey.
All right, thank you, Hans. Our next presenter is Dr. Susanna Kulla, head of the Applied Alternative Methods Group at BASF in Germany. After working for BASF as a postdoc in the field of alternative toxicological methods, Susanna became responsible for BASF's Laboratory for Alternative Methods Development in 2007. Her tasks included the establishment and validation of alternative toxicological methods for a variety of endpoints. Since 2009, Susanna has been heading BASF's Laboratory for Applied Alternative Methods, a testing laboratory dedicated to routine testing using alternative methods for testing skin and eye irritation, skin sensitization, respiratory toxicity, and endocrine disruption. Take it away, Susanna.
Thank you very much, Mandy. So I would like to introduce to you today the background review document prepared by ICFAM. And, well, we will not use any time, but start straight away. On this very first slide, I have given you the reference if you need any further information. So this is a thick document, so I've just pulled out a few information for you. So sorry, this was too quick. Okay, so this ICFAM evaluation took place after the EPA nomination of the BCOP as an ocular irritation test in 2003. The purpose of the background review document was to describe the current validation status. This included the accuracy and also the reliability of the BCOP method, amongst others. Also, it was evaluated how broad the scope of the substances tested was, as well as the viability of standardized test method protocols. The performance analysis was performed on 18 peer-reviewed publications. Those publications included BCOP test method results, as well as protocol information. And also 5 studies were what we nowadays would call validation studies comparing in vivo and in vitro data. Furthermore, unpublished information on 3 additional studies were submitted to NICETAM, and in total then 8 in vitro and in vivo comparative studies were used for the analysis of the assay. so. Talking about the protocols, I will not go in the very details here. This is just to summarize that there were some common principles in the assays. These included the isolation and culture of the corneas, as Hans has introduced to us just a couple of minutes ago. Well, all protocols included a test substance treatment, and the endpoints determined were opacity as well as permeability. This were the common principles. On the other hand, there were variations in the protocols. Those included the instrumentation for the opacity determination, the exposure concentrations and durations, and the prediction model used. Coming back to the exposure concentrations and durations, Hans has already introduced to you that there's different protocols used for different purposes, and this has also been the case in the history of the assay. Sorry, this was too quick. So this slide is introducing to you the different instruments that have been used in the background review document studies. So again, we show here the opacitometer from Spectro Designs. So I've listed here some technical information on the device. So the opacitometer is using a white light. It does have two light sources for zeroing. Generally, it's a center-weighted reading of the cornea. And it's used, it's calibrated using the three filters, so in a linear range. So this is the details of just one available opacitometers. In the literature, different devices have been used, including a UV spectrophotometer, as well as laser-based opacitometers. Hans has nicely introduced the calculation of the in vitro irritation score, so we don't have to talk about the details here. So again, for the OECD guideline protocol, we're using opacity and permeability values only, so histology is currently not considered. In the table down here, I have summarized the OECD, the classification laid out in the OECD guideline, and this is based on the GHS category. So according to the OECD guideline, the BCOP is used to differentiate between compounds that are corrosive or severe eye irritants, so GHS category 1 chemicals. And on the other hand, we have non-corrosives or non-severe, while we cannot differentiate currently between the category 2As, 2Bs, and not to be labeled compounds. Historically, on the other hand, and this is what is shown here, different classes have been defined and are currently under discussion on an OECD level. So subclasses included non-eye irritants, in an in vitro irritation score range between 0 and 3, above 3 up to 25 for mild irritants, and 25 higher than 25 up to 55 for moderate irritants. And in accordance with the OECD guideline, anything with a score higher than 55 would be classified as severe eye irritant. So without going into too much detail, I would just like to bring to you that during the ICFAM validation, different chemical classes were evaluated. Major classes included alcohols, carboxylic esters, carboxylic acids, esters, formulations, heterocyclic compounds, and so on. Due to the development of the assay and the use in former times, different product classes have been assessed. Those included chemical and synthetic intermediates, cleaners, drugs, and pharmaceuticals, and so on. So in total, 161 substances and formulations were included in the evaluation of the BCOP in the ICFAM documents. Here we have summarized the performance. So as with other in vitro methods, the typical parameters that are being evaluated are the accuracy, so the overall concordance of the assay compared to the in vivo results. As listed here, approximately 120 of the 150 compounds were correctly predicted, so this makes an accuracy of 81%. We had a sensitivity of 84% and a specificity of 80%. So all in all, this, the predictivity values of the BCOP very, very satisfactory and very promising. There's a but to the story, and the but to the story is the overprediction rate. So an overprediction in the BCOP would then lead to a false classification, so a compound that is not a severe eye irritant would end up in a GHS category 1. In total, 20% of the compounds were falsely predicted. And coming back to the chemical classes, and as has also been introduced by Hans, alcohols, ketones, carboxylic acids, and heterocyclic compounds were often overpredicted. And talking about the physical form, liquids did have a higher likelihood to be overpredicted compared to solids. The other value we have shown here is the underprediction rate. So this is severe eye irritants that are not correctly predicted by the BCOP, and those included alcohols, solids, rather than liquids. And due to the nature of the assay, compounds causing persistent lesions in the in vivo assay were more frequently underpredicted than those causing reversible lesions. So in summary, the discordant classes of in the BCOP included alcohols, ketones, and solids. So this was my summary of the ICFAM documents. In the next couple of slides, I would like to show to you the evaluation of the different opacitometer models that we have performed during our in-house validation and establishment of the method. So I would like to start this with the pictures of our instruments. So we can see here now 2 opacitometer models. One that is a little bit older one on the left side, and the instrument on the right is technically identical. Only the luminometer is included in the instrument. So we have in the housing we have here this black little hole that is the size of a cornea holder, so that's where we place the cornea holder in. We have a light source on one side of the instrument and the light measuring detector on the other side, and we can also read our opacities on the screen here. The whole instrument is connected to computer, so there's no need to note down the values manually. So in our comparison, we have also summarized the technical information of the instruments that we have compared. So this is again the instrument of electro design. We kindly were provided with an instrument by IRES to do these evaluations, but we have also evaluated an instrument from lab research. Then we had our own prototype as well as the current technical status of the instrument. So there were differences in the light sources as well as the light detectors, but well, we now ended up with a halogen lamp and a light detector that is commercially available from instrument producers. On this, well, rather complicated looking slide, we have now compared the opacities that were obtained with what we now then assumed as a standard opacitometer and our instruments. And we can see here, so the different colors is the different instruments that have been used. And in all cases, we can see that we have a rather linear relation between the 2 instruments. So we have the standard instrument here plotted against the new instruments on the other axis. And as high opacity values are, so very high opacity values don't really make a difference. If you recall the prediction model, we have done the evaluation for corneas with an opacity value smaller than 100, and in total we have included in this evaluation more than 60 corneas. Yeah. So this slide is summarizing the evaluation of the instruments and showing you the mathematical relation between the values, because the OECD guideline refers to the standard opacitometer opacity, but we have then determined using all these corneas the evaluation how to actually obtain the opacities according to the OECD guideline using our devices. So this slide is showing you the number of corneas that have been treated, the correlation factor, the correlation of all treated corneas and those with smaller opacity values. And in the last column, we have shown you the algorithm that is used to calculate the opacity values. So here we have another graphical representation of the correlation of the opacity values. So we have tested here the proficiency chemicals as have been listed in the OECD guideline. So you can, well, they're abbreviated, but we can see the compounds down here. And then we have used the standard opacitometer as well as our opacitometer and calculated the opacity values. And comparing the two values, we can see that the standard deviation of the individual opacity values from the data shown here is smaller using our opacitometer, but in general the opacities are very comparable. So the same or similar experiments have been performed at IIVS to see whether our equipment works also in a different laboratory, and this is shown in this graph. So IIVS has also done an assessment of the BASF equipment compared to the standard equipment, and you can see here that we have a correlation of 99% here. So in the next couple of slides, we will move to an application of the BCOP assay. So we have in the past assessed a lot of agrochemical formulations, and what I want to show to you here is the application and the data of the substances. To start with, we have assessed 50 agrochemical formulations that had available in vivo data, so we did not have to do any additional trace testing. So this was a applicability domain specific validation performed at our lab. To start with, because we've done this assessment in the light of different classification schemes, this slide is summarizing the formulations that have been tested, and this is showing you that the classification systems are similar but not 100% alike. So we have 9 formulations that are GHS category 1 and also EPA category 1. We have a good correlation in the middle categories, but there's a bunch of formulations that are not classified according to GHS but have EPA category 3. So this is just to give you an overview of what type of formulations have been tested. The formulations in general are multi-component mixtures that are made for pesticide purposes. So we have solvents in there and of course a lot of active ingredients. So in this slide, I have summarized the data on the 50 formulations tested. We have 2, so the stacked bars are showing you the opacity values in blue and the 15 times the permeability in orange, and this is to give you the in vitro score in total. So how to read this slide? So all category 1 chemicals, if you recall the prediction model, should have in vitro iteration scores above 55. So from just the prediction model, we would expect all formulations that are here in the GHS1 category to end up with high in vitro irritation scores. But this is unfortunately not what we found. On the other hand, all the formulations that are not to be categorized according to GHS or are GHS categories 2B or 2A should be found below this cutoff of 55. And what we can see is that all non-irritant formulations were actually found between below the cutoff of 55, but not the ones that are in vivo category 2s. So this is the OECD guideline endpoints. In addition, we have performed histology on the corneas after the standard endpoints in the BCOP assay. So without going into too much detail, we have a scoring. We have developed our internal scoring system for the pathophysiology of the bovine corneals. And 0 would be a cornea that is not affected at all, and the 5 is the most severe category. And as you can see from this summary, it looks like the histology has a quite good predictive capacity. So all the category 1s are more severely affected in terms of histology grade than the ones in category 2A, 2B, and the ones that are non-irritant according to GHS are found in the very low categories. So, and to end this presentation, I have summarized here the BCOP data on the 50 formulations. This is box plots, so this is giving us an interpretation of how predictive a single endpoint of the BCOP is. So again, we can see here that there is no no clear correlation of the opacity value to the GHS category. The same is true for the permeability value, so there's no clear trend of the permeability value to increase with GHS categories. Histology, as we've just seen, is predictive for the severity of eye irritation. And I think we're now coming to my last slide. That is summarizing of how we use the BCOP. So we have a two-step strategy. So as we've learned today, the BCOP alone is currently not regulatorily accepted, not even in Europe, to assess eye irritation as a standalone assay. So we use the BCOP to test for severe eye irritants. So if we have a positive result in the BCOP, we end up in the GHS category 1. If the BCOP, on the other hand, is negative, further testing is needed. And what has been established at BASF is the epiocular test. So that's a reconstructed corneal model that is then used to filter out the negative, so the non-irritants. And if the epiocular is positive, we have a mild irritant. So this strategy works both ways. Well, depending on what you expect your material to be in terms of irritation potency, you can start either way. So now I think with this slide, I would like to thank you very much for your attention and hand back over to Hans.
Thank you, Susanna. Hans has some additional information on the use of the BCOP assay for materials falling into the category of antimicrobial cleaning products.
So I'll give a quick brief on the history of this project. Several years ago, there was an interest among a number of manufacturers of hard surface cleaners to recognize and to market the recognition that these hard surface cleaners are actually quite effective at antimicrobial activity as well. Here in the United States, any claim on a product of antimicrobial activity for a cleaning agent falls clearly into the EPA's antimicrobial cleaning products labeling program and has classically required animal eye irritation testing to evaluate and label the eye irritation potential. And since many of these products have already been successfully marketed on the market for, in some cases, several decades prior to making an antimicrobial claim, the thought was among the stakeholders, the industry as well as folks at EPA that we're not introducing a new product. We're essentially adding a claim, and that perhaps there's a non-animal way to evaluate for eye irritation potential. And so this drove a project to look at the BCOP, as well as two other assay systems, the three-dimensional human corneal ocular model, as well as the cytosensor microphysiometer, to look at the potential for those assays to label for eye irritation. And so on the screen here, the first pass was to look at a series of chemicals and formulations, should I say, with known EPA category history from the Drays test, which you see on the abscissa, with categories 1, 2, 3, and 4, and test those products in the BCOP using the standard 10-minute exposure protocol. And what we see here is that on first pass, there is a large group of materials here that were EPA category 1, which were giving in vitro scores of well above 75, certainly above the severe threshold of 55 originally established. There's also a nice population of non-irritants on the EPA category 4 level here as well. Now, we recognize that the BCOP has the potential for inaccurate predictions in the EPA category 3 and 4 category. So the thought was to couple the BCOP with other assays, such as the three-dimensional human corneal model, the epiocular, to look at the irritation potential for those milder materials in those other models, such as the epiocular. We recognize also that for EPA category 1 that there are several formulations here that were, at least these two here, that were potentially underpredicted and labeled as category 2. So one of the goals here then was to see if we could utilize the histopathology as yet a third endpoint to evaluate those materials that were underpredicted for severe categories. And this model comes from Jim Maurer and Jamie Jester with the concept that they were able to identify that the initial injuries in the rabbit cornea were highly predictive of the likelihood for recovery downstream in the Drays test, such that materials which are non-irritant or slightly irritant typically involved only the rabbit corneal epithelium and were likely to recover relatively quickly in less than a week or so. Mild to moderate to severes had deeper and more pervasive damage into the cornea, and the depth of those injuries, as well as the nature of those injuries, were then predictive of whether there are corneal erosions and whether the recovery was likely. And certainly for severe irritants that were penetrating and damaging halfway into the cornea were unlikely to recover in the 21-day test and hence would be labeled as severe. And using this concept then, we looked at the histopathology for those corneas treated in the BCOP assay with these antimicrobial cleaning products and gave also a rough assessment of a suggested EPA category and the types of damage and lesions that we saw and the depth of those lesions into the cornea, such that we found that among those products where we only had cell loss in the upper or mid epithelium, those were likely EPA category 4s. Those which just extended perhaps into the upper stroma were likely EPA category 3s, and so on and so forth. And of course, the severes were those that caused damage well into the stroma of the bovine cornea in the BCOP assay. And so using that information then, we went back to look at how one might modify the prediction for these same materials and recognize for the aggressive materials here that were underpredicted that we got an appropriate correction to category 1. These two were still at category 2. We had a number here that were clearly predicted as category 1s. We took a risk that for some of these category 4 materials that there was some potential for over-predictions. But having said that, in the EPA antimicrobial cleaning products program, we used a tiered approach with the top-down approach using the BCOP first to look at strong or severe irritants or EPA category 1 or 2 materials to label for EPA category 1 or 2. If those materials were not found to be strong irritants by the BCOP, then we would evaluate correctly with either the cytosensor microphysiometer assay or the epiocular to label for category 4 or 3. In a similar fashion, the bottom-up approach was that if those latter two assay systems suggested a severe irritant, then we could appropriately label using the BCOP assay, whether the materials were EPA category 1s or 2s, where we actually have quite nice predictions and a relatively low likelihood of under predictions using that assay system. Okay, well, thank you for your attention.
Hey, thanks, Hans. We will now open up discussions to address some of your questions. We have a couple minutes. The first question is, how do you apply viscous materials? Hans or Susanna?
Okay. In one of the slides, we showed the rinsing of materials and the application materials by applying very fluid materials directly into the port in the bovine cornea holder chamber, and basically being able to rinse those materials also using those small ports. For viscous materials, we need to use different tools. We have positive displacement pipettes, which allow us to pipette accurately very viscous materials. And the approach we take is something called the open chamber window technique. This is actually described in the OECD guideline. I'm glad this question came up because I'd like to clarify that this is simply a means of removing the anterior chamber window, the glass circular window from the holder, removing all media in the front chamber, and applying the viscous materials directly onto the cornea and getting corneal coverage. And those chamber windows can be replaced back onto the cornea, incubated, and at the rinsing step, we again remove that chamber window and rinse by gently rinsing a stream of media directly onto the into the chamber and using a swirl technique to rinse out the test chemical. Simply an easy, effective way to remove viscous materials.
Okay. There's a question that came in. Please tell us the vendor for the opacitometer. So, Susanna, this is for you.
Well, yeah, thank you very much for the question. So when we started to establish the BCOP in our laboratory, well, we experienced that it's not very easy to obtain an opacitometer. And then what we did is we rebuilt one, and we actually now sell it. So to make the technology available to people that want to establish this method. So if you want some further information, just write me an email and I'll get a quotation to you.
Excellent. Here's one to Hans. How do you proceed with solid compounds which comes to a paste after it's diluted with water or saline?
So this is a material, if I understand correctly, that has essentially solidified to a paste material. That can be quite a challenge. Perhaps in many of these cases where we have these challenges, we certainly want to have a good discussion with the sponsor to understand the nature of the testing. The nature of the exposure modeling is always critical to modeling that in vitro. There are potential approaches that might include further dilution of the material as a liquid. Alternatively, we might envision testing the solid directly and not trying to make a 20% suspension, and perhaps testing the solid directly onto the cornea. These are activities that sometimes need to take a bit of customization to address these unique situations.
Okay. How can a new lab begin running the BCOP assay?
Susanna, would you like to say?
Yes, because actually we had so great teachers to do so. So first of all, what we did is, well, we studied the literature as we would for any new method that we want to establish. But then we found a laboratory that has the capacity to do the assay. And kindly enough, IIDS helped us with the training. So we had actually IIDS come over to Germany and train our technicians to perform the assay. Then, of course, we experienced, as I've mentioned already, we experienced difficulties with the equipment, but found a solution to ourselves. And probably the most important thing then is to show proficiency of the laboratory. And the way we did this is we tested a variety of chemicals. So in total, we had around 60 chemicals that were tested. most of them listed in the OECD guideline as proficiency chemicals, but we also have included some of those listed in the ICFAM background review documents. On top of this, we have tested BASF internal chemistry just to see whether, so first of all, to show our proficiency, how predictive the assay is, and in particular, how predictive it is for our real-life chemistry.
Very good.
Okay. If alcohols are over and under predicted, why is ethanol used as a positive control?
Well, our experience with ethanol has been that we obtain a chemical which has a very reproducible opacity and permeability impact on the corneas. Our goals for using a material as a positive control are really to assess the ability of our execution to be reproducible, to assure that the equipment is performing properly, to evaluate the quality of the eyes. And so we use that positive control really as our measure for our reproducible conduct and execution. As it turns out, the ethanol in the 10-minute exposure gives results that fall essentially on the moderate to just below severe prediction in most cases. And the in vivo data actually from the S-talk suggests essentially the same result, that there are data showing that this is a moderate irritant and occasionally becomes, or so occasionally it's severe in a couple of animals.
Okay. What could be the approximate cost to standardize technique?
Yeah, so well, it very much depends on where your starting point is. And well, most laboratories do have a photometer, so to measure this permeability endpoint. And the well, approximate cost of the positometer is around 12,000 euros. And this comes then with the opacitometer, including the corneal chambers, filters to calibrate your device. So that's everything around the opacitometer. And very shortly, we will also be able to include in this kit a video to show you how to perform the opacity measurements and the corneal preparation and all that.
And I guess the second part to that question is developing the capabilities and the proficiency to conduct the assay. And Susanna's group probably also recognized that the ability to become proficient in the assay and to understand some of the nuances probably is measured in a couple of months worth of time of direct hands-on activity.
Okay. Why are the concentrations of fluorescein different for solids and liquids?
I think that
That's an interesting
came
one.
back from the, yeah, and they are very similar, aren't they? 0.4 and 0.5%.
Yeah.
Essentially, it's a bit anecdotal. It comes historically from evaluating the predictions of the series of liquids at Merck and the series of solid intermediates, and the 0.5% used for solids just was sufficient higher concentration to get slightly higher optical density values to get those solids in the appropriate prediction category relative to the in vivo data they had at hand.
Okay, and I think we'll do one last question. Please expand on the 0 to 5 scoring scale for histology. I believe that one's to you, Susanna.
Ah, yeah. So, well, as we have introduced to you, current endpoints of the OECD guideline are limited to opacity and permeability. But what we know, well, due to experience as at IIVS or also at BASF, is that histology presents a useful endpoint. So the histology is not standardized currently, so that's an activity that's ongoing. To align the bovine corneal pathologists worldwide to use a similar grading scale. So I can just briefly explain to you what we do is we process the corneas, just like Hans so during our routine pathology processing, and then we have a pathologist looking at the slides. And behind all the scoring, there's a lot of factors that are being assessed. So it's the degree of damage to the corneas in the summary, but we actually look at the stroma, we look at the endothelium, at the epithelium. We try to investigate the corneal swelling and all that. So it's a very so the whole score is just a single number, but behind the scenes we have at least 10 points that are being evaluated for the pathology, and well, in my opinion, and I'm not a pathologist, it does take some expert training to be able to evaluate these corneas.
Okay. I'd like to thank all of our panelists and all of the attendees for your participation in the webinar today. We hope that you've gained a better understanding of the BCOP assay, its uses and limitations, and the current regulatory status. We would like to remind you that any questions not answered during the Q&A time will be addressed by the panelists, and the answers will be disseminated to all of the webinar participants, along with a PDF of the presentation slides. We're planning to host our next webinar in April, and we will be discussing the cytosensor microphysiometer assay. Please check our webpage, www.iivs.org, for more information after the new year. Thank you once again for your participation, and we hope to see you again soon at a meeting or during our next webinar.